Question number 106. In animal cells, steroidal hormones are synthesized by RAR, SER, ribosomes or Golgi bodies. As you know that endoplasmic reticulum are of two types, RAR and SER. RAR is involved in protein synthesis, whereas SER is involved in lipid synthesis. Steroidal hormones are lipids in nature. So RAR, no. Ribosome help in protein synthesis. This cannot be a correct option. Golgi bodies, they do not synthesize anything. They help in modifications of proteins and lipids. So this is not the correct answer. Second, SER. Yes, SER or the smooth endoplasmic reticulum helps in synthesis of steroidal hormones. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 107. Match the following columns and select the correct option. Column 1 shows the different types of chromosomes on the basis of position of centromere, and column 2 shows the shapes of these chromosomes during anaphase. So you can analyze in this figure that these are different chromosomes in the metaphase and these are their shapes during the anaphase. Here you can see that this is metacentric chromosome and it appears V-shaped during the anaphase. You can see here it is sub-metacentric chromosome as centromere is just away from the middle and this appears L-shaped. This chromosome is acrocentric as centromere is present near end and in anaphase it appears as J-shaped. This one is telocentric and this appears as I-shaped. So you can analyze that metacentric, V-shaped, telocentric, I-shaped, acrocentric, J-shaped, submetacentric, L-shaped. So A goes with second. So these two options are ruled out. B goes with third. This option is ruled out. C with fourth and D with 1. By analyzing all the options, we can say that the second option is the correct one. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 108. Spherical structures present in nuclear matrix and are site of active rRNA synthesis are absent in Chlamydomonas, amoeba, purple and green photosynthetic bacteria, a plant cell. This question is asking about the spherical structure present in the nuclear matrix and is the site of rRNA synthesis. In the nucleus of eukaryotes, there is a spherical structure present called nucleolus. Nucleolus is the site for rRNA synthesis. It is the spherical structure. As nucleolus is present in the nucleus and nucleus is the monopoly of eukaryotes. The question is asking about the in which it is absent. It must be absent in prokaryotes then. Chlamydomonas, a green alga, means eukaryotic. Nucleolus will be present. Amoeba, a protozoan, a eukaryote, it will be present. Purple and green photosynthetic bacteria, no, they are bacteria and bacteria are the sole members of kingdom Monera, means they are prokaryotes. So nucleolus will not be present here. A plant cell, plants, kingdom Plantae, yes, they are eukaryotes and nucleolus will be present over here. So the correct option for the given question is option number 3. Let's move on to the next question. Question number 109. Identify the structure indicated by label A in the given diagram and choose the correct option regarding it. First, spherical structure called kinetochore. Second, site of attachment of spindle fibers. Third, primary construction called kinetochore. Fourth, disc-shaped structure which prevents chromosome shortening or loss. So you need to identify this structure, the structure which is labeled as A, and then choose the correct option. With the structure which is labeled as A is nothing but kinetochore, which is disc-shaped structure. So, spherical structure called kinetochore? No, it is not the spherical structure. It is a disc-shaped structure. It is site of attachment of spindle fibers? Yes. Kinetochore is the site of attachment of spindle fibers during cell division. Primary construction called kinetochore? No, kinetochore is not the primary construction. The primary construction is centromere. Fourth, disc-shaped structure which prevents chromosome shortening or loss. It is a disc-shaped structure, but it does not prevent chromosome shortening or loss. Chromosome shortening and loss is prevented by the end part of the chromosome, which is called telomere. So the correct option for the given question is option number 2. Let's move on to the next question. Question number 110. Chromosomes sometimes have a small fragment separated by non-staining secondary construction called chromatid, satellite, centromere, centrosome. Here you can see this is the structure of a typical chromosome in which two chromatids are shown. This is one chromatid and this is the second one. Each chromosome has a primary construction called the centromere. Sometimes some chromosomes have secondary construction also. Beyond the second construction, the part of the chromosome is known as satellite, as shown in this figure. So this is chromatid. This is secondary construction, and beyond the secondary construction you can see the part of the chromosome which is satellite. The secondary construction does not take the stain, it is the non-staining region. The primary construction is known as centromere. What about the centrosome? Centrosome is the cell organelle which contains centrioles. So, the chromosomes sometimes have a small fragment separated by non-staining secondary construction called chromatid. No. Centromere, it is the primary construction. Centrosome, no. So the correct option for the given question is option number 2. Let's move on to the next question.